Hi, and welcome to the next webinar in our Plantae webinar series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you're experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you have questions for our panelists during today's webinar, please let us know using the GoToWebinar chat box. We will moderate these questions and read them aloud to our panelists. This webinar series is brought to you today by Plantae, the open online community for plant scientists, powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today to support this series and use the discount code webinar10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. Be sure to follow us on social media. The hashtag for this series is Plantae Webinar. In response to the COVID-19 pandemic, many organizations are canceling, postponing, or moving their meetings online. There have been many new online seminar series and journal clubs initiated by our community to stay connected with each other. Check out our global calendar to browse all of the opportunities in one place. This calendar is collaborative, and we encourage you to submit plant science events of your own. All right, thank you for joining us today. I'm now going to turn it over to Dr. Rob Furl to kick off introductions and moderate today's questions.
Good day, everybody, and thank you for joining what is going to be, I think, a very interesting discussion about both career paths and unusual career paths for plant biologists in our world, and quite honestly, not in our world. The way in which your panelists found their way to becoming plant biologists in the space biology business are as varied as any sort of career paths and trajectories you might find in science. And we come to you today as a group of people that, well, quite honestly, orbit Kennedy Space Center in one way or another, associated with the process of, well, using spaceflight to better understand plants here on Earth, or using plants to better enable spaceflight by eventually supplying carbohydrates, fresh air, and water to our colonists on another world, or even our astronauts on the International Space Station. So plants offer a really interesting opportunity to sort of engage with space biology for those two very important reasons. One is that there's a lot of things we'd like to know about plants that are gravity driven, and being in space allows us to take gravity out of the equation. And the other, the sort of more fundamental one for exploration, is the fact, quite simply, that if we're going to live on another world for any period of time, we're going to have plants with us, and those plants will be helping with our oxygen, water, and food. The people on your panel today each represent some aspect and some different aspect of those particular topics having to do with plants in space. We are going to go through a series of introductions and talk about our career trajectories that brought us to this point, and then open it up for questions to the group as a whole. So my name is Rob Furl. I'm a professor at the University of Florida, and this is my introduction slide. It is an expression of, in my case, a very traditional trajectory of academic science. Got my PhD in biology and gene expression, and my interests at the time were in using environmental stress as a way to understand how genes worked, to understand what gene expression meant and how it happened in the world of plants. So I started out in the business trying to understand how genes work using plants, and in fact, using plants that were underwater and stressed as a way to get at the questions of how genes work. During the space shuttle era, as plants came back from space, some of them actually looked like they were suffering from being underwater or expressing some of the same phenotypes as plants under stress. So that dragged me, pulled me, and excited me on the idea of using spaceflight to better understand gene expression and using my tools for gene expression to better understand how plants can be adapted to spaceflight. I'm a professor at the University of Florida, do pretty traditional professorial types of things. Our next panelist is Gioia Massa, who's a life sciences project scientist at Kennedy Space Center. And the next slide will introduce her and her career trajectory.
Thanks, Rob. So yeah, for me, I got really interested in this field pretty early on. When I was in 7th grade, I had an agriculture teacher who was invited out to Kennedy Space Center to learn about the work going on to grow plants for food. And he brought back that information, and it really inspired me. So throughout junior high and high school, I built hydroponic systems and got really excited about this field. I went to college and I was working there in a plant hormone lab for Peter Davies' lab. While there, I also got to come to Kennedy Space Center as an intern through the Space Life Sciences training program. So that was a real boost up to encourage me to stay in this area. For graduate school, I went to Penn State and I studied with Professor Simon Gilroy, who's a good space biologist, very prominent space biologist as well that we all know. And he worked on gravity signaling. And I was really interested in how plants might use gravity signaling in kind of a real world application. So how plants responded to obstacles. So this sort of central figure is from one of my PhD studies on looking at root responses to gravity and touch, what happens when they hit an obstacle. And while at Penn State, I got to be involved in a student spaceflight experiment on the space shuttle, looking at seed germination. So that was another sort of plus up for my career along the way. I realized that I wasn't really as interested in the fundamental signaling as I was interested in kind of the food production side of things. So next I went to Purdue University to have a postdoctoral appointment with Dr. Carrie Mitchell as part of this NASA Specialized Center of Research and Training in Advanced Life Support. And so I got to work here on crop plants, including strawberries, and a lot of work on LED lighting. And if we go to the next slide, my journey continued at Kennedy Space Center. So we can go to the next one, where I was also a postdoc. I came as a NASA postdoctoral fellow to work with Ray Wheeler, who you'll hear from in a moment, and studied horticultural optimization of the veggie hardware for the space station. This was before it flew. I was hired on by NASA and was able to continue the veggie work to do the first flight tests of veggie as the science team lead, which also led to some interesting experiences like being able to go to the White House and plant some of the veggie plants in the White House garden. More recently, I've been a PI on an investigation, and you can see in the top right corner where Jessica and Christina are running an experiment that we're looking at for food crops in response to different wavelengths of LED lights. But the most fun I think I have is down in the bottom right, where I work a lot with students and teachers, and this is just one of the programs that I'm involved with, where for this Growing Beyond Earth program, we have over 230 middle schools and high schools around the country helping us to select the new crops for space. So that's probably my favorite thing to do. Thanks.
Very good, thank you, Gioia. Our next panelist is Howard Levine, who's the Chief Scientist at the National Utilization and Life Sciences Office at Kennedy Space Center. Howard?
Thanks, Rob. Next slide, please. So I guess similar to Gioia, around 7th grade, I decided I really wanted to be a marine biologist. And I ended up getting a zoology degree for my bachelor's. And then for my master's, I worked at, and this is the upper left side of the slide, the Aquacultural Engineering Lab in Wareham, Massachusetts, where we had different kinds of pilot plants for growing oysters. And while growing oysters in a flow-through raceway system, I began to study what they were eating, and I ended up taking a phycology or algae course, and I became fascinated with the seaweeds. So below that, you see a picture of sea lettuce, ulva, and I ended up using ulva as a biomonitor of pollution in coastal waters. I would both collect natural populations and grind them up and look at what pollutants they had accumulated. And then I developed a method for seeding their spores onto little wooden sticks that I could then deploy at selected locations. After my PhD, I went on to do a postdoc at the Marine Sciences Research Center at State University of New York at Stony Brook. And this was a marine biomass project where we were growing kelp to eventually digest down to methane or ethanol. So that was a lot of fun. We had a flow-through seawater greenhouse system that we did a lot of phenotypic plasticity work in. And then eventually we had a farm that we deployed in Long Island Sound, and just to see if we could practically grow kelp large scale. And then in the upper center, eventually, of course, the biomass project went away when the energy crisis went away, and I was hired by Professor Abraham Kokorian at the Department of Biochemistry and Cell Biology at SUNY Stony Brook, doing tissue culture, plant tissue culture. And he had a spaceflight experiment. It was actually the first life science spaceflight experiment that was managed out of Kennedy Space Center. And we were able to grow plants aseptically, tissue culture derived and sterile seedling clones, and bring them back and look for chromosomal aberrations in the root tips. And we actually had 3 of those spaceflight experiments, after which I was hired at Kennedy Space Center as a project science coordinator. And during that time, I was awarded a grant to do a spaceflight experiment using porous tubes, comparing plants at 3 different wetness levels on porous tubes, as well as substrate nutrient delivery systems. And then subsequently, there was an opportunity to actually do the first plant experiment on the International Space Station. This is on the upper right. It's called the Jose experiment, Jason Outreach Seed Experiment, in which corn and soybean seeds were germinated. But in general, I had the opportunity to work with multiple principal investigators on spaceflight experiments, somewhere between 70 and 80, and ranging from sea urchins in the Aquatic Research Facility. I was chairman of the Institutional Animal Care and Use Committee for 14 years, so that we reviewed and oversaw all rodent experiments that went up to the International Space Station. One of the more interesting experiments was called CEBUS, the Closed Equilibrated Biological Aquatic Systems, which had swordtail fish and snails and an aquatic angiosperm and a microbial filter, and it was a recycling system, actually a bioregenerative life support system that flew in space. And recently we have the Advanced Plant Habitat that is up on the International Space Station, growing plants larger scale. And currently I'm trying to validate a new way of using a nutrient delivery system in the veggie system that Gioia mentioned previously. So that's it for me. Thanks, Rob.
Very good. Thank you, Howard. Next up is Ray Wheeler, who on my screen at least, is the Andy Sugar with the bookcase behind him. Ray is a plant physiologist, and Ray, you're located at Kennedy Space Center for a long time, and the court is yours. Can't hear you, Ray, sorry.
Let me unmute first. Thank you. Can you hear me now okay?
Yep.
Okay, sorry. And I apologize, Andy, for thank you for getting me linked in. I'm sorry, I'm an imposter posing as you. Yes, my name's Ray Wheeler, and the next slide, I think I did my slide a little bit different than some of the others, but hopefully it can show you the path that I followed in terms of getting toward where I am now. I'll go way back to my days as a Boy Scout. This was really important to me as an individual in terms of really getting an appreciation for biology and life sciences, the opportunity to see living plants and animals and nature. So that had a profound impact on me as I went to college. I initially, my BS is actually in astronomy, so it didn't affect me there, but it kind of came through as I had opportunities to get into graduate school. And I had a chance to work with a person named Frank Salisbury. I don't know if that name means much to some of the folks listening in, but Frank was, and his colleague Cleon Ross, co-authored plant physiology textbooks that were used around the world for probably 20 years. And so that was a great privilege to be able to work with Frank at Utah State. And he happened to have a grant from NASA. So that got me connected with some of the life science activities that NASA was sponsoring at that time. And in particular, I was looking at plant gravitropism, specifically with stems, and trying to understand some of the mechanisms there. And that was really a neat experience. It got me connected with the NASA space biology at that time called the gravitational biology community. Then, like all of us, I think probably on the panel today, we couldn't get a real job right away, and so I went on to become a postdoc, which is a real job, and you really work hard. But it was a great step for me. I went on to the University of Wisconsin, and so I had a chance to work with a fellow named Ted Tibbetts, who's really a horticulturist, horticultural physiologist. And so this was a little bit of a change for me in terms of the emphasis of my work. And I studied growing plants for life support mission in space. This is something that Rob mentioned up front in his introduction. And in particular, I focused on growing potatoes in controlled environments, and kind of ironic that that's what the Martian Matt Damon ended up growing in Mars. I think he picked a pretty good crop, to be honest. But it was a great experience for me. It taught me a lot about how controlled environments can be used to do research with plants. And then that gave me the opportunity then to apply for a job at Kennedy Space Center. And at Kennedy Space Center, it was just a fantastic experience. That was 1988 when I started there. And we had some very unique chambers. One was a very large chamber that actually had vertical shelves of hydroponic systems and light banks. And to my knowledge, it's probably one of the first working examples of a vertical farm. But it was a closed chamber, and it allowed us to do some very unique measurements of canopy or community gas exchange rates. And by that, I mean tracking canopy photosynthesis, respiration, transpiration. And we did this for a range of crops. So that was just a great experience. And that's I'm still at Kennedy Space Center today. I'm a senior scientist in our life sciences group. I've sort of broadened out over my career and gotten a little bit more into life support systems that include not just biological approaches, but so-called physicochemical approaches too. So the next slide was really, when Rob asked me to put together these slides, it was something that I thought was important for me to do, where I just sort of wanted to point out people that were really important in my professional trajectory. And I just want to emphasize the importance of finding good mentors and having good colleagues that you work with. It really helps you through your career. It certainly did for me. And I wouldn't be where I am today without the help of all these people. So thank you.
Thank you, Ray. We are going to now pull away from people that are NASA civil servants in the plant biology business and back towards people that are academic scientists associated with Kennedy Space Center and space biology. Our next panelist is Dr. Annalisa Paul, a professor at the University of Florida and director of the Biotechnology Center there. Annalisa? Can't hear you, Annalisa.
Aha, I was muted, sorry. All right, so as Rob said, I'm an academic scientist. Came up through the ranks in your standard academic model. Where I came from is a little bit different. I did my doctoral, first I did my master's work in two kind of disparate things. One was in marine botany, and so from your data now, you might say that, oh yeah, working with algae is probably a good pathway, but statistically probably not. But I worked with algae and marine algae and physiological ecology. So from that perspective, I was very interested early on in how the environment impacts the structure and function of plant parts and plant biology. I also worked in the biochemical realm, looking at different type of secondary compounds in citrus and other plants. And I'm a hardcore botanist. So I was educated in botany and plant physiology and plant molecular biology. For my doctoral work, I work mostly in gene regulation from the perspective of chromatin structure and DNA architecture. So quite different from the physiological ecology realm. But when you put everything together, it actually predisposes one quite nicely to go into a career of space biology. So if I go on to the next slide, what that shows is sort of a synopsis of all the work that I've had the honor to be a part of in the last 20 plus years. And so going from a background of where you want to understand how plants respond to environmental stress, space biology, the spaceflight environment, is the coolest, most novel type of outside the realm of evolutionary experience environment that you can imagine. And so the work that I've done in the last 20 odd years has been doing just that, is finding out from the molecular level how plants respond, what genes are induced, what genes are repressed, what kind of biochemical changes happen, what kind of cell-specific information can we get by using fluorescent reporter genes. I've used a number of different tools, a number of different approaches, including most recently epigenetic approaches, looking at how the architecture of the genome changes in response to spaceflight. I've also done a lot of work in what we call analog environments. And so there's spaceflight analogs for things like suborbital research or parabolic research, where you're doing experiments in short-term transitions into microgravity, but also taking all of that curiosity and knowledge into what do we need to know before we go to another planetary surface, and practicing some of those things in greenhouses, both in the high Canadian Arctic and also down in Antarctica, where we can ask, all right, so now that we're learning about how plants respond to strange and novel environments, how can we test some of the things that we've learned, and how can we reveal some of the things by going to these remote environments that we wouldn't really necessarily uncover by working in our own backyard. And so that's pretty much me in a nutshell.
Thank you, Annalisa. And our last panelist is the real Andrew Sugar, the guy with the desk behind him. Andy is also a professor at the University of Florida, but he has an interesting distinction from the rest of us in that his laboratory and office and research environment is located at Kennedy Space Center. So he's a hybrid academic person being right there in the middle of the mix at Kennedy Space Center physically. So Andy?
Okay, thanks. Hello, everyone. Thanks for chiming in. If I could have the next slide. I became interested in plant biology in high school in my advanced biology class, and I had an opportunity to work as an honors student in a plant pathology research lab in a nearby botanical garden. And that started my interest in plant pathology. I eventually went on to college and got a series of degrees in plant pathology. The latest was a PhD in plant pathology at the University of Florida. But this first slide actually shows my my sort of working career start, which was at the Epcot Center's agricultural pavilion called The Land. And there's two images there. One in the upper left shows a rather large research plot that we actually did in the greenhouses, looking at plant disease development on various plants. In this case, it was wheat and beans. And that was what I did at Epcot in the Land Pavilion, is just basically work on how to diagnose diseases. And then when we had a persistent disease that was affecting a plant in sort of a major way, I would then be responsible for working with the entire staff and coming up with a control program. So the first part of my career out of college was about 16, 17 years of just traditional plant pathology, which I really enjoyed. And then in 1996, there was a news conference at the Johnson Space Center about 5 scientists reporting in this news conference that they believed they had found evidence of extinct fossilized microbial life in a Martian meteorite. The Martian meteorite was recovered in Antarctica, and then the description of these structures was done at the Johnson Space Center. And I thought that was so incredible that right there at that news conference in the summer of 1996, I decided I wanted to go into astrobiology. Next slide, please. So I started the transition into astrobiology, specifically focused on Mars, because, one back, hopefully we didn't delete it, one back. I started to get interested in Mars because that was the location or the planetary body in our solar system that probably has one of the best chances of finding an extant, viable microbial community present in the lithosphere, or the upper crust of the planet. So humans would like to explore Mars. There's a lot of robotic missions and orbiter missions going to Mars. So of all the planetary bodies in the solar system, that's the one I became the most interested in. And so I've been working for about 20 years on the survival, growth, and adaptation of terrestrial microorganisms to Martian conditions. But the real long-term goal has always been, even from 1996, is to get prepared for helping to support human colonies on Mars through helping to develop bioregenerative life support systems. And as a plant pathologist, my interest in those is to help protect the plants from the development of diseases that might occur in transit to Mars, or when a colony is set up on Mars, trying to protect the crop cultivars that are required and necessary for food production from potential microbial pathogens. Next slide. And then the last thing I wanted to mention is that if you're all thinking of graduate school, you want to try to pick some of the really coolest and most interesting field work that you can get your hands on. And this is an experiment I did. The red pod hanging off of this jet flying over the Kennedy Space Center has got the abbreviation DART for DART, and it stands for Dust Atmospheric Recovery Technology. And in the jet, in the back seat, is me at about 15,000 feet, collecting aerosol dust, looking for plant pathogens that might be transported in atmospheric dust from Africa. So it's a real diverse, eclectic kind of background, but it all is focused on microbial survival and adaptation to very low pressured environments. And that continues from high altitude sites on Earth through the atmosphere of Earth and then on to other planetary bodies. So with that, I'll sum up and say I look forward to your questions.
Well, thank you, Andy, and thank you all for your description of your journeys. The key, or one of the key things we wanted to talk about today and to let people know about, is that there's no single phenotype for a plant space biologist. We all look different, we all come from different backgrounds, and we all find ourselves here because we are interested in space flight and space biology, but also because our scientific backgrounds simply brought us towards these kinds of questions. And I'm going to split our online questions up into a couple of different topics and sort of bounce between the two topics. One is, how did we get there, and what a career trajectory might look like for us, as well as some of the basic questions people are asking about living and working in space and the role of plants in those particular things. I'm going to start off with a very quick question because it follows a couple of the questions, and that is, are there future careers in plant space biology? And I think I'll direct that question to Gioia and Howard and Ray Wheeler, who are right there at Kennedy Space Center and see the trajectory of plants in space at the current time.
Okay, well, I'll just start and let Howard and Ray answer as well. I think there are definitely future careers. You know, there are so many questions that we have yet to answer in terms of, you know, the fundamental plant responses to the space environment, but also the applied ways to grow food using plants, using other organisms. So I think, you know, a lot of these questions are really critical to answer to enable human exploration. And so there will be opportunities both in academia, in industry, and in government.
Thanks, Gioia. I'll just add to that in the past 5 years or so, NASA has really started to get serious about plant production in space. And I think part of the reason is because we're going back to the moon and then on to Mars. So currently what we do is we mostly bring all of our food with us, kind of like going on a picnic. But as you have longer and longer duration missions, that becomes less viable. So that, for instance, whenever you see a moon habitat, you usually see a greenhouse attached to it. So I think that we're in a time when there's increasing emphasis on crop production in space, both on planetary surfaces and actually in microgravity, as you would need for the transit from Earth to Mars.
And Ray, you got anything you'd like to add to that?
I agree with Gioia and Howard's comments. I think there are opportunities. Agencies like NASA, you know, the opportunities kind of wax and wane a bit. But I'll give you an example. In the 1990s, probably when we had our peak staffing for research at Kennedy Space Center for our controlled and bioregenerative life support systems, we probably had 5 or 6 plant biologists working in our team, 3 or 4 microbiologists, 4 or 5 agricultural engineers, some chemists, so it was a diverse group, but there were clearly a lot of opportunities, and it was a great experience for me. So I think there will be, as Howard mentioned, and Gioia.
Very good. I'm going to switch now from the future to a little bit more on the past and ask both Annalisa and Howard to talk about the diversity of their sort of science trajectory coming from, if you will, coastal ecology into plant-space biology. Can you guys reflect a little bit on intentional changes in your career trajectory as opposed to just what happened as you sort of walk down the path?
All right, I'll jump in first. For me, I grew up in Florida. So I grew up being a science geek from a very early age, and also being very attuned to the space program. So I always had it in the back of my brain that I wanted to be a scientist ever since about the 3rd grade, but never thought that I would be a space scientist per se. I was very interested, as I mentioned, in sort of the environmental effects that the conditions had on plants, and how you could have the exact same species of plant but have actual different responses to that depending on the environment that you lived in. So for me, I always wanted to know how plants work. And so that's why I started as a botanist, then went to be a plant physiologist, and then a biochemist, and then a molecular geneticist, trying to go ever deeper into the why and how question. And so for me, the trajectory was, the door was opened to explore a truly novel environment, and that helped me answer my desire of how and why plants work. And also because I've always been like I said, a science geek, a science fiction fan, and I wanted to be a part of what took plants off planet and allowed us to understand how we could grow them in space and on a colony in Mars. And so that was the motivation and sort of the trajectory. Not totally deliberate, but certainly aligned.
Howard, anything to add to the coastal discussion?
Yeah, usually when people ask me, what should I do if I want to become a space biologist, I tell them, well, get the best science background that you can. You don't start studying space biology, you study, botany or zoology or whatever research area you're interested in. And although I was also a science fiction geek, and I always really was interested in the space program, I never really envisioned that I would end up as participating in it. And it was fortuitous because, as I was saying before, I was working on a marine biomass project. The money went away. I transferred to someone who, like Ray, happened to have a NASA grant. And that was how I entered into, the space biz, as I'll say. So I think the key point there is just get the best basic background in the sciences that you can. And then when you reach a certain point, you can begin to look for space-related options if you're so inclined.
All right, I'm going to switch to a very practical question that has appeared online, and that is, are there jobs in space biology open right now, and where does somebody go to look for those jobs? Any of you NASA people got an answer for that?
Yeah. I mean, I can say that for the NASA position, there's a website, USA Jobs, where all NASA positions are advertised. There may be additional ones at the universities for professors with research grants looking for grad students or postdocs. So those are the different categories. And of course, there's more and more companies that are getting involved in space research. So there's a series of those that can also offer job opportunities.
The other thing I'll mention as part of the job and career track discussion is the American Society for Gravitational and Space Research, ASGSR, which has a very active student chapter and a very active community of people that look for students and postdocs all the time. So that society is one you might want to get into if you are interested in this kind of career path. I'm going to shift the question.
Rob, and that website is www.asgsr.org.
Yep, very good. I'm going to shift the question over to Andy Sugar and ask him to reflect just a little bit more on the notion of, on basically two notions, how your experience working for a company, basically working for the land and for Disney, how that that obviously didn't get in the way of your trajectory to the space business, but it is unique amongst the group here. Can you reflect on going over to basically industry for a while?
Well, yeah, that's a really interesting question. I had a phenomenal opportunity working at the Land Pavilion at Epcot Center because in addition to being the senior plant pathologist on the staff there, I was also tasked with interacting with NASA, scientists out at the Kennedy Space Center, and I was given the freedom to attend many workshops early in the career. And I've seen some of the questions as we're going through here that kind of talk about how do I get started in this kind of any subtopic of this field. And the best way to, in my view, is try to go to either a national or international conference that includes some aspect of space biology, listen to the talks, and the talks that really get you fired up and excited, try to go up and speak with the speaker, either right after their talk or try to look for them during the poster session. And so it's networking that really gets you those opportunities that you don't really know are out there. In my situation, I met Ray Wheeler and Howard and everybody else here years and years ago. In fact, Ray started working at Epcot started working at NASA just a few years after I worked at Epcot, and we just naturally started talking and working together and communicating. So it's that networking that really helps you out. So if you're interested, try to attend a topical workshop, symposium, or conference. It's the best way to get started.
Okay, I'm going to shift the topic just a little bit to a question for Annalisa and Ray, having to do with there's a comment online that a lot of the work happening these days in space is with model systems, but what sort of research pathways for actual crop plants exists or has existed? And I think what I'll do is I'll ask Ray to talk a little bit about the history of actual crops in space and in space-related science, and then ask Annalisa to reflect on model organisms and their interactions with those. So Ray?
Well, NASA has sponsored, by the way, other space agencies as well, but NASA has sponsored a lot of work with crops. Most of that has more typically been what's referred to as ground-based research, because the thinking was that the use of crops for space missions would really begin to trade more favorably as you got to surface destinations like Mars or the Moon. But there's a continuum there, and so crops have a role even in early missions, and Gioia is involved with a lot of this research right now of just providing supplemental foods. So there tends to be a focus right now on leafy greens because they fit well in spaceflight plant chambers. They're relatively easy to grow. There are challenges in space. So things like lettuce and some mustard greens and Zuna and things like this have been tried, and we're hoping to get to things like small fruiting crops, tomatoes and peppers. So these aren't really model crops per se, or model organisms, you might say, but they're kind of jumping ahead to the application side of using plants in a space setting, and they can yield good research. You know, if you design your experiments and ask good questions as you approach these things. But I'll defer to Annalisa on model organisms. She has, and as well as Rob, have a lot of experience there.
So, and I've also sort of been looking through some of the questions. There's been a lot of questions on genomics, on DNA structures, on gene expression and stuff. And the answer to that is, yeah, a lot of that work has been done with model organisms. And the model organisms that are most commonly used are ones that have their roots, if you will, in edible things, like in the mustard family, Mizuna, Arabidopsis. All those things are have edible cousins. And so the things that we learn by using model organisms that are small and easy to work with and have got short life cycles allow us to extrapolate into how the crops that we might want to take with us would be better suited for spaceflight and planetary environments. And so in other words, the things that we learn about how model organisms adapt to spaceflight environments allows us to either breed, engineer, or select varieties of plants that are edible and more suitable for surviving in these kinds of environments. It's a huge part of spaceflight research.
So I'm going to kick it one time over to Gioia. If you could take a minute, you look over and watch over and have seen the sort of long-term development of the veggie growth unit for the International Space Station, and you've seen it look at, grow, and take care of many kinds of species. Can you just sort of, on your fingers, run down the number of different plants that have grown in space inside of veggie?
Sure. We've been focusing from a food side primarily on leafy green crops so far in veggie. We've grown about 8 or 9 different varieties of leafy greens, lettuces, Mizuna mustard, Chinese cabbage, red Russian kale, and wasabi mustard is a really fun, spicy one. We also grew zinnias, which were, a flowering crop in preparation for testing small fruiting crops like tomato and pepper. Then, of course, we've done a lot of model organisms. So Arabidopsis, as you and Annalisa are very well aware, has been grown quite extensively in veggie, along with Brachypodium. And the Europeans have actually had an educational payload where they tested lentil, mustard, and radish seed germination in veggie, which was pretty fun. We've also had algae growth in veggie in bags specifically designed for that. So it's just designed to be a versatile platform. We hope to start growing dwarf tomatoes in there next year, and peppers at some point as well.
Very good. Andy, I'm going to get a question over to you, and it's a general one that basically has to do with the focus on soil microbial communities, microbial life on other planets. This is from a Canadian listener, but the question I think is universal, and that is, talk to us a little bit about the relationship between the microbial survival that you've looked at in your chambers and indeed in places like the Canadian Arctic, and the relationship between that and plant health.
Right. So of all the work that's been published on how microbes might survive under Martian conditions, the most dominant biocidal factor on Mars is the solar UV irradiation. There's no ozone in the Martian atmosphere, or let me rephrase that, there's only a trace of ozone in the Martian atmosphere, and it occurs only very periodically. So we don't get that an ozone layer that's attenuating the strong UVC and lower UVB photons. And so the environmental condition on the surface is actually much more biocidal on Mars than it is on Earth, even though Mars is twice the distance. But if a microbe is transported to Mars inside a human habitat or microbial community, is transported to Mars inside a human habitat, those individual microbes and the communities are going to be protected from that solar UV irradiation. And they will likely perform in much the same way as they would here on Earth in any other form of hydroponic or soil-based agriculture system. I mean, Mars has a lower gravity. It's only 0.38 Gs compared to our 1 G, so there's going to be an effect there. But other than that, inside a human habitat that will have to have an atmosphere that can support humans and pressure and gas composition, microbial communities will likely perform very similar to what we see on Earth. The big difference is ionizing radiation that is not filtered out by the thinner Martian atmosphere. And some of those ionizing radiation forms can get through the outer layers of space habitats. And so that might be a factor that would alter these plant microbes interactions a little bit more.
Very good, thank you. I'm reflecting on another question that's come in here recently that perhaps we can not clear up, but continue to comment on a little bit. And that is that Ray mentioned that the peak of plant physiology jobs at Kennedy Space Center was in the 80s or 90s, and Howard says that new jobs are opening up. So can you NASA folks sort of help the community out there understand where, you know, sort of in the grand scheme of, you know, not necessarily comparing to the heyday, or are we entering the heyday, a new heyday of when plant people will be in the exploration business, not just working at KFC, but around the world? That's for Gioia, Ray, or Howard.
Well, I'll start. You know, I mean, Ray and I kind of laugh because we've been in this business for about 30 years and always pushing what we refer to as bioregenerative life support systems, where, you know, you grow plants to scrub CO2 and produce oxygen and clean water. And oh, by the way, they're also producing food. And NASA has always said, yeah, you know, that's great. You know, we got to get around to that maybe in about 5 years. But it's always been 5 years away. But the change that happened about 5 years ago, and a large part of this is due to Gioia's effort at getting funding from the Human Research Program, is that they've decided that now is the time. So, and also in the Space, Life, and Physical Sciences division that those of us working for NASA at KSC are within, they are putting great emphasis on crop production. So there is in general a ramping up on the part of NASA in the interest in growing crop plants for food in space right now.
Yeah, if I can add to that, the one thing I really believe is going to happen over the next 10 years or so is that with the industrial partners in the world, companies like SpaceX, Blue Origins, many others who have come out publicly and actually are spending a lot of time and money developing their own private industrial capabilities of sending humans to the moon, which is the goal of Blue Origins, and of possibly then also sending humans to Mars, which is the stated goal of SpaceX. The physics doesn't change regardless of who is going to these planetary bodies. If NASA is going to be the primary lead, or Blue Origins or SpaceX, whomever, it's almost likely to be a consortium, but whomever the collection of organizations, they're going to need life support systems to support and allow humans to flourish in those environments. And one of the real key things is plant production in these what are called bioregenerative life support systems, because if you can use plants to recycle oxygen and water and produce food crops, there's a real cost benefit of not having to ship those resources to the planetary bodies. So don't just look to NASA or academia for careers in plant biology. look to these other aerospace companies that are very aggressively pursuing very similar goals.
For one question back about career trajectories and for our international audience, I think I'm going to ask Ray to comment upon opportunities, I don't know, basically around the world, non-US opportunities in space biology. You're familiar a lot with the European and Russian connections, and if you could make some comment there on the international sort of state of affairs, and then hand off to Gioia or Howard for additional stuff worldwide, that'd be great.
Sure. Yeah, the notion of using bioregenerative approaches for life support for space exploration is really a global concept, and the first researchers that were really pushing this hard were Russians in the 1960s, and there was activity in the U.S. as well, mostly focused on algae at that time, single cellular algae, chlorella. But through the years, European groups got interested. The European Space Agency and most recently the German Space Agency is really engaging in a lot of ways. The Japanese Space Agency has had space research funding for probably 30 years, and they built a large demonstration facility in northern Japan to actually test out the concepts of bioregenerative life support with humans. So, and now probably the most recent or the newcomer to this is the Chinese National Space Agency. And they're sponsoring a lot of bioregenerative life support work that involves controlled environment agriculture systems with humans and waste processing systems. So there is certainly global interest, and for our Canadian Space Agency as well. And a lot of us on this panel have interacted with colleagues at the University of Guelph in Ontario. They've been doing a lot in terms of very innovative and sort of trailblazing work in terms of understanding plants and how they can be used for space exploration.
Gioia?
Just to follow up, I guess, you know, I know some of the students are asking, are there opportunities for international students? The only opportunity at Kennedy Space Center is through the NASA Postdoctoral program, which does allow international postdocs to work with NASA. But international students can certainly work at university labs where, you know, investigators have space biology research programs as well.
Very good. Katie, I can turn the screen back over to you for winding up the hour, if you like.
Certainly. Thank you all for such a great conversation. This is really cool stuff that I wasn't even aware of. We do have on Plantae a spot to continue to have these conversations. So if you join the Space Biology Network on Plantae, there's different collections related to this topic, and also a place for you to ask more questions and answer them with the community. So I really encourage you all to join that network and start up some discussions. We weren't able to get to all of the questions today, so we will be posting the follow-up questions on this site. So be on the lookout for that.
And Katie, if I could add one sort of last bit before you finish up with us for today.
Sure.
I want to express my sort of sincerest wish that anybody who has tuned in today and is interested in space biology to maintain that interest. Find a way to stay involved, first off, and then think about the many opportunities people have talked about today, the many opportunities that appeared in people's career paths that got them towards this business and that are in it today. And think about it, if you are, just sort of keep that in the back of your mind as you move to your various companies or classes or postdocs or degrees or any place you go. There are ways to keep this interest burning. I can tell you from the large science perspective that interest in the space program and interest in space-related science is growing now. We now have at the International Space Station something called the International Space Station National Laboratory, which gives another non-NASA way to get experiments into space, especially for private industry. So you may find yourself in any one of a number of different venues that can point towards space as a place to do experiments. Katie?
Yep, very well said. I think earlier today it was also mentioned that networking is a big component of advancing your career. And through our online community and also through this Plantae Mentoring Center that we've recently started, those are a great way to build connections in the absence of being able to travel to meetings right now. So definitely stay in touch and reach out to people in this way. You can volunteer as a mentor or as a mentee by following this link. And lastly, don't forget to join ASPB. ASPB membership dues are what makes these webinars possible and open to everyone. So be sure to join, and thank you all for attending. Thank you especially to our panelists for having such a great discussion. We'll be following up with additional questions here soon and posting the recording.
Thank you.
Bye, everyone.